So, thank you for everybody for coming to this week's Cell Size and Growth Seminar Series. We're really excited to have Vicki Loscik from Boston College and Kesuke Kuromiya from Stanford University, who are both going to speak to us about the role cell size and cell size change plays in various um, organismal physiologies. So thank you, Vicki and Kuro, and take it away, Vicki. Thank you.
Thank you so much to the organizers for having me here to talk about my work. I'm just going to share my slides and set up before I forget, the laser pointer. I think that's working. Okay. All right. Yes, it's great to be here and be able to talk to an audience that cares so much about cell size. My lab has been interested in studying tissue repair for a long time because we have a lifetime of tissue damage to endure. And when tissues are injured, you think of cells being lost here at the site of injury, and that these cells have to be regenerated or repaired in some way. And many tissues rely on cell division through stem cells to recover cells that are lost. But what we've learned in the last decade, that in addition to cell proliferation, cells can also grow in size by becoming polyploid to repair the damage. And here we're defining polyploidy as any cell that has more than the diploid copy of the genome. And polyploidy contributes to repair and regeneration of a number of tissues across different animals. I think this was first most eloquently demonstrated in the liver, where hepatocytes become polyploid to help regenerate the liver. Polyploid cells are seen to rise in the kidney in response to acute damage, also in the heart and in the epicardium of the zebrafish during regeneration. You can see multinucleated polyploid cells form at the leading edge. And we also see this in the fruit fly, which is what my lab uses as a model to study polyploidy in tissue repair. Fruit flies are a great model system, so I'll introduce this as well for the other speaker. To use, they have a short generation time, breed large number of animals, and most importantly, we have exquisite tools to control gene expression. The one that I'll be using throughout my talk is the Galvor-US system, where we can express genes of interest in the epithelium, either knocking down genes or overexpressing different mutants or markers. All right, to study polyploidy in the adult fruit fly epithelium, we don't yet have a live imaging technique available. We dissect out the adult fruit fly abdomen, cut it open, pin it to a Siligard plate, fix it, and then do immunofluorescence. So all the images that you're looking at today are from different flies, but we can overall see the trend of how these polyploid cells form in response to injury. First part of my talk, I'll be talking about how we use an insect pin to injure the fruit fly, which creates a wound scar. In my second part of my talk today, I'll be talking about other injury through UV irradiation. Okay, but when you immunofluorescence stain the adult epithelium, which is on the ventral side, this is what the epithelial cells look like. And you can see that the epithelial nuclei here are labeled in green and the junctions are magenta. This is a postmitotic monolayer of epithelial cells, and the epithelial cells are diploid. When we injure the epithelium by puncturing through the outer cuticle with an insect pin, you lose about 100 cells, and in its place, by 3 days, you get a giant multinucleated polyploid cell that I've outlined here in this dotted yellow line. This giant polyploid cell forms through two mechanisms. One, an incomplete cell cycle known as the endocycle, where cells go into S phase, bypass mitosis, and then go through a gap phase, and also through cell fusion. And these two mechanisms to make this large cell are happening at the same time, and you actually have to inhibit both the endocycle and cell fusion to prevent polyploid growth, which is shown here. And when you do that, you can see that the wound cannot close, and this demonstrates that polyploidy is required for tissue repair. So after observing this as a postdoc, and when I started my own lab, I really wanted to determine what were the physiological functions of polyploidy? Why do you need these polyploid cells to heal tissues like in the fruit fly when it's damaged? And we learned a lot about the physiological function of polyploidy. It actually has a number of beneficial roles. These polyploid cells can compensate for cell loss without relying on cell division. They're more resistant to DNA damage, and they prevent healing in the presence of genotoxic stress. And they're also necessary to restore mechanical tension to the abdomen after injury. And since doing all those studies, which are down here, we've been more interested in the last couple of years about trying to understand what are the signals that initiate polyploid growth, and how is polyploid growth, contained? How do the cells know how much polyploid I need to restrict to heal the wound? So these are the two stories that I'll be telling you about today. First, on what's necessary to regulate the stent of growth, and then what's necessary to initiate growth in response to injury. All right, so we had worked out some of the signaling pathways that were important in polyploid growth over the years. It includes the hippo signaling pathways for activating Yorkie, which is required for regulating G to S phase genes. They're necessary for cells to go into the endocycle. And we also found that RAC GTP is necessary for cells to fuse. And upstream of this is integrin. Integrin is a cell adhesion which interacts with the extracellular matrix. Okay, so at the wound site, we know that there's really a lot of remodeling of the extracellular matrix and formation of this melanin scar. So a grad student at the time, Luis L. Gonzalez, in my lab, asked, does scar formation at the wound site regulate wound-induced polyploidization? So let me just tell you a little bit more about this melanin scar that forms in Drosophila. Melanization happens very quickly after injury, within 1 hour, and it serves a role in the immune response to encapsulate microorganisms. It releases reactive oxygen species, and is speculated to function in wound healing. So when you have injury, you get a proteolytic cascade that activates this prophenyl oxidase and cleaves it into phenyl oxidase. And in our system, we could actually answer whether this scar formation was necessary for wound healing or not. We can express membrane RFP in the epithelium and see whether the wound is able to heal. And we can prevent the flies from making melanin at the site of injury by simply taking mutants that lack all three phenyl oxidase genes. So that's shown here. You have the wild type fly, you wound it, you get a melanin scar at the site of injury, and you express a membrane RFP. You can see that the epithelium is able to close the wound underneath that scar. And if you look at a phenyl oxidase null mutant, you don't get melanization at the site of injury, and the wound remains open. So we're able to confirm what people speculate in the field, is that this melanin scar is necessary for wound closure. And we saw a similar response here. If we look at our polyploid mutant that endocycle can't fuse, you also see the wounds open. So we initially thought that this melanin scar was going to be a signal to initiate polyploid growth. But then when we looked at these mutants that can't make the melanin scar, we actually saw the opposite of what we predicted. So, and that's shown here. So here in the wild type, at 3 days post-injury, we see this giant multinucleated cell forms, okay? And in this mutant, this melanization mutant, you get a little bit of autofluorescence here. I think it's from the clot, but you get this giant, much larger multinucleated cell form, as outlined by this yellow dot line. Okay, and if we quantify the size of that multinucleated cell, this is known as a syncytium, we can see that those cells are indeed much larger when we don't make the melanin scar. We also notice that the nuclei were getting really huge in these mutants as well around the wound. You can see these giant nuclei in comparison to the wild type. So we measured the DNA content of the nuclei, and in the wild type here, prior to injury, you can see that the nuclei are diploid. After injury, about 30% of the nuclei around the wound become tetraploid. When we don't have these enzymes that make melanization at the site of the wound. The epithelium develops normally. It's still diploid, but after injury, you can see there's an exacerbated polyploid response, and the nuclei reach higher ploidy, and there's more nuclei that are responding. So we suspected that this was due to a change in the cell cycle, and we looked at the cell cycle by looking at flies that had PCNA, which is a protein that's important for cells to go into S phase of the cell cycle. And here you can just see a time course in the wild type. And we knew from previous studies, and we show here as well, that epithelial cells around the wound enter S phase about 24 hours post-injury, and this continues until 3 days post-injury or 72 hours post-injury. Now, when we look at the melanization mutants that can't make that wound scar, we see that, all right, my screen keeps moving in and out. I see that these epithelial cells around the wound now go into the cell cycle much earlier. So at 12 hours post-injury, we see about 100 nuclei around the wound are going into S phase. And then by 24 hours, it's even more magnified. And we see that there's up to 400 or more nuclei responding to this wound. So this is really quite dramatic. It says that the scar is necessary to restrain the polyploid response post-injury. So that sort of concludes the first part of my talk. This work is published in Genetics, so you can read more about it. But what we suspect is that this melanin scar that forms is necessary to restrain a signal that's emanating from the wound site that's necessary to trigger these cells to endocycle and fuse. And when you don't have that scar, the wound stays open and the signal persists and it's able to spread, and the whole tissue is responding to the injury now. In addition, we're looking at more of the mechanism to figure out how the scar is regulating the genes we already know they're important in polyploid growth. In the paper, we showed that junk signaling and June was misregulated, so that's contributing. We suspect the hippo pathway is also misregulated as well, and we're continuing to look at that in my lab. All right, now I want to switch gears and talk about another story that has to do with the initiation of polyploid growth. And this is completely different. Okay, so I'm sorry, these are two kind of different stories, but I hope it gives you kind of a flavor of what my lab is working on. This paper, the story I'm going to tell you about, is available as a preprint, and it should be in press in PNAS any day now. Okay, so it's known that genotoxic stress or DNA damage can lead to polyploid growth in cells that are going through the mitotic cell cycle. Okay, so you have some sort of genotoxic stress from the environment, like radiation or gamma radiation or sunlight, like UV, that leads to DNA damage, or you can have replication stress. And if a cell is going through the mitotic cell cycle, that will lead to G1 arrest. They'll become tetraploid. And then if they're able to bypass that arrest and bypass apoptosis, they'll continue to endoreplicate. And as cells endoreplicate, they're also known to be more resistant to genotoxic stress. But we wondered what happens in cells that are not cycling, that are post-mitotic. If they encounter genotoxic stress, are they also going to be able to become polyploid, and through what mechanism? So to induce genotoxic stress in the fruit fly epithelium, we expose them to UV irradiation at a low dose of 25 millijoules. We just stuck the fruit flies with their sort of bellies facing up into a Stratogene oven with the UV light. All right, and we first checked, this work was again done by another grad student in my lab, Ming-Chi Shen. We first checked that UV radiation induces DNA damage by staining with a phosphohistone H2A antibody, and phosphohistone is known to bind to sites of DNA damage, so it's often used as a marker for DNA damage. Here, the epithelial nuclei are in green, okay, and I'm pointing to the epithelial nuclei here. The muscle nuclei in this tissue always have DNA damage, all right? So in uninjured tissue, there's only a low-level occasional cell that has DNA damage, but after UV, we can see much more frequent DNA damage within these epithelial nuclei, as pointed out by the arrowheads. And you can see if you measure the intensity of H2AV, you can see that that's significantly increased at 2 days post UV irradiation. Interestingly, if we look at the DNA damage over a long time course of more than a week post-injury, the DNA damage in these epithelial cells persist. And you can see that these epithelial nuclei greens still have high levels of DNA damage. So what's happening to the epithelium? Now we're UV irradiating the epithelium instead of poking it with a needle to see what's happened and whether polyploid cells are induced. Okay, so we have the uninjured tissue here, 0 millijoules, looks similar to before with the epithelial nuclei in green, junctions in magenta. 2 days post UV irradiation, we start to see some cell loss. So some of these post-mitotic cells are dying post UV. And at 5 days, that's where we see the greatest cell loss. So it sort of bottoms out about 50% of the epithelial cells will die in response to the UV irradiation. But the ones that are able to survive, they start to remodel their junctions at 7 days. And you can start to see more continuous junctions here. And also here, I just showed the junctions alone in this bottom image. And by 10 days, you can see that the tissue is repaired by making these giant multinucleated cells similar to what we saw with a puncture wound. You can also see that some of these nuclei have really increased in size. This is a giant one that we found here. So we went through, I'm not showing all the data here, but we were able to confirm that in response to UV irradiation, the epithelium has a very similar injury response where the cells are able to survive by both entering the endocycle and by fusing to repair the tissue. And we were wondering, well, which of these mechanisms is really important for cell survival and repair after UV irradiation? So we used our Gal4U system to inhibit the endocycle. And we just confirmed that here. You can see that if we measure the nuclear ploidy of the epithelial cells post-injury, we see about 20 to 30% of the nuclei become polyploid. When we knock down E2F1, that blocks the ability of cells to go into S phase, that increase is inhibited. Okay, but then we look at the overall tissue morphology, we can see that these cells are still able to fuse, they're still able to repair the tissue, and we don't see any enhanced nuclear loss. So the endocycle appears to be dispensable for tissue repair post-UV irradiation. Next, we looked at cell fusion, and we saw something quite striking. Okay, here's the wild type again. You can see that mono and multinucleated polyploid cells form. And when we express RAC GTPase dominant negative, this is a regulator of cell fusion in our tissue. We see only mononucleated cells here on the periphery, but then in the center of the tissue, we find that there's no tissue. The cells are lost, they're dying. All right, there's just areas devoid of nuclei, showing that cell fusion is necessary for tissue repair and cell survival post-UV. So we were wondering what was happening to lead to this response. So of course we did a time course. I'm just showing you instead of 7 days, 3 days post-injury. And if you look at this RAC dominant negative, the tissue develops normally, but we can see enhanced apoptosis throughout the tissue. When we look closely at this, we can see that here's a dying cell with the condensed nuclei. We're getting mononucleated cells are not able to fuse, so they're not able to repair the tissue. If we look at the wild type condition, here 25 millijoules post UV, we can catch some cells where we notice that there's a dying cell and the junctions are starting to remodel around these dying cells. So there seems to be a connection with cell death and the ability of these cells to fuse. And this is actually not surprising because in development, like in osteoclasts that make up the bone, osteoclasts become multinucleated during their differentiation. And it's been shown that there's a link between cell fusion and apoptosis. The signals that are important that usually are associated with apoptosis, such as caspase activation and phosphatidylserine exposure, are also necessary for these osteoclasts to fuse. And if you inhibit these processes, then the osteoclasts cannot make multinucleated cells. So we wondered if we could inhibit caspase activation similar to what's been done in osteoclasts in our cells and see what the consequence would be. So we used this baculovirus protein P35 to block caspase activation. And first, we confirmed that this would inhibit cell death, and it did. Okay, so this is looking 7 days post UV here. This is without UV, post UV. And we can see these giant multinucleated polyploid cells arising in our wild type. And there's about 50% cell loss. And when we look at our epithelium expressing P35, where caspase is inhibited, we see fewer cells are lost. And we also see that these polyploid cells are not forming. Okay, we don't see enlargement of the epithelial nuclei. Like you can see these big polyploid nuclei arising, and we don't see that when we inhibit caspase. We measured DNA content to confirm that. We see that there isn't as high of a nuclear ploidy when caspase is inhibited, as shown here in the graph. And then we also wanted to see if fusion was effective. We did see that there was sort of remodeling of these junctions here. They looked a little bit different. They're a bit harder to keep track of, so we just had to use another method to see whether fusion was changing. And to do that, we use a mosaic labeling technique where we express two different colors within the epithelium. And if those colors mix, that's indicative of cell fusion. And you can see here that prior to UV irradiation, the colors are separate, but post UV irradiation, you get merging of the colors. And in the P35 expressing epithelium, we can see that these colors remain separate and there's not spreading of the EGFP area, indicating that fusion is also inhibited. Okay, so going back to our original question, what are the signals that initiate polyploidy? We found that genotoxic stress through UV irradiation can induce polyploidy through caspase activation in postmitotic cells, and it leads to cells endoreplication, but it also leads to cells fusing. And unlike in mitotically dividing cells that endareplicate, and then that endoreplication is important in resistance to genotoxic stress, we find in this system that the resistance to genotoxic stress is dependent on cell fusion. Okay, so just to wrap up here, some remaining questions that we have are to really try to figure out more of the mechanism of what are the genes within this apoptotic caspase pathway that are required for polyploidization, both endocycle and cell fusion. And we're really interested to see whether this is conserved in mammalian postmitotic cells that also become polyploid. All right. And with that, I'll just wrap up and take any questions you may have. I'd like to thank the grad students and the undergraduates that worked on this project, as well as my funding from NIH and NSF. Thank you.
Thank you so much, Vicki. That was beautiful. I will, as always, there's lots of questions already in the chat, and we'll go through them. I have a question to begin with, which is kind of going back to the melanization story. The kind of the runaway uh, peri-wound growth really reminds me of something that like a SASP-like response, and whether I'm wondering whether you see any sort of cellular senescence phenotype developing at all in the wounded tissues.
Yes, Brian Calvi's lab has done some really nice work on that in oogenesis, and we haven't yet looked at markers, but after his study came out, that's something that I've definitely wanted to do to see when these cells keep endocycling, do they eventually senesce, or are they, because they're still cycling much longer, they stay within, they're not senescing yet? That's a good
Yeah.
question.
All right, great. Good. Yeah, looking forward to hearing about that. First we have a question from Shyama, who doesn't have a mic, so I'll ask her question. So she wants to know, do the PPO null flies experience more DNA damage at the wound site?
We haven't looked at that, but that is a good question that we could definitely check for. It could be the case with the DNA damage that persists, when cells endocycle, there's a lot of stalled forks, and that can create DNA damage itself. So there could be DNA damage just from the endocycle itself. So that's something to consider when cells are endocycling.
Okay, thank you. Next we have a question from Dave.
Thanks so much for your talk. It was beautiful. So I couldn't help but think of another epithelium in mammals, melanomas. So do you think melanin is actually a response to proliferation, an attempt to inhibit it in melanomas?
I don't know. I mean, I know there's a lot of connections to cancer, and the melanization pathway does share some of similar genes because it forms through dopamine synthesis. But I don't know. I haven't looked at that aspect at all. So that would be definitely something to consider whether there's a connection. I mean, I think there's still a lot of polyploidy to be identified out there, and often, labs don't consider whether their cells are polyploid or not.
Thank you.
Thank you. Next, we have a question from Kiki.
As wound healing progresses, does there occur a transition eventually back from multinucleate cells to cells with a single nucleus? If yes, how does this physically happen, especially considering how the nuclei appear to be unevenly distributed across the syncytium in your images? If no, what happens instead?
So it really depends on the tissue and organ cell type. So in the flies that we study, the polyploid cells are permanent once they form and they never revert back to their diploid state. I know one example in the epicardium where they saw those multinucleated cells form, they apoptosed and were replaced by dividing cells in that example. So I think it really depends on the tissue, whether the polyploid cells, you know, persist or are replaced by diploid dividing cells.
I could imagine a rather ridiculous implication of this if in some tissues there really is no transition back to normal mononucleate cells. If you kept damaging the epithelium in an expanding patch, could you conceivably convert large areas of the fly to multinucleate cells? And if so, what would happen? For example, are those cells functional?
Are you saying if I injure the tissue multiple times, would I be able to
You
convert?
injure one spot and then let it form multinucleate cells, and then injure the spot beside it, and so on. Would you end up being able to grow a very big patch of these multinucleate cells because they never turn back?
Yes. Yes, you can probably, I mean, the UV is kind of like a big damage because the whole epithelium is, we're putting these flies in the UV oven and their whole epithelium is being damaged and we get a huge wound. So the whole epithelium pretty much fuses together, as I show with that rainbow analysis. And we also know that with age, the epithelial cells, they don't endocycle, but they spontaneously fuse with age. So the whole epithelium can become polyploid, and we're really interested in using this model to understand when your tissue changes from being diploid to polyploid, how does that affect physiological function? In the
Does
wound, it seems to be beneficial. Long term with aging, we have seen some detrimental effects where it reduces the mechanical tension of the epithelium. Does that
in
answer your
Sure.
question?
does the fly's
Okay.
body still work?
Sorry, sorry,
Yeah.
Kiki. Let's move on to the next. There's a lot of questions in the queue. Um, we'll go to Mike next.
Hi. That was awesome. I was wondering if there's a long-term in the cells that I think when you irradiate them and they're able to fuse, but you inhibit endoreduplication, right? You have the big fused cells, but you have many fewer genomes because
Yeah.
they are not, they're not endocycling. Like what happens? Is there a like what happens in the
Is there
long
a
term?
long-term impact? We haven't looked past 7 days. We do know the endocycle is necessary to restore, like to compensate for the cell loss. So we do these experiments where we measure the total ploidy within the tissue. And if they don't endocycle, they can't restore the ploidy they're still able to heal by fusing together, and we don't know what the long-term impact of having a epithelium with half the number of genomes, but that is a really interesting
Cool.
question. They seem to be fine. Uh,
Okay.
Fortunate for us, the epithelium is like when our GAL4 driver turns on after these cells have divided and differentiated, and we can like actually, express an apoptotic gene and just kill the epithelium, and the flies seem to be fine in the laboratory. So the epithelium is not essential for survival in the lab. It's just the ventral epithelium that we're destroying, not the other epithelial cell types.
Okay, thank you.
Yeah.
Okay. Let's just take one last question from Lucy O'Brien, and there's two more questions in the chat. If you have time, Vicky, could you kind of
Or
reply to
I
them?
will.
Yes, thank you so much.
Um, Thanks, Mimi, and thanks, Vicki. That was really, really cool. Especially love the gorgeous rainbow data. Um, Simple question: have you ever played with painting the abdomens of these UV irradiated flies, or like putting black tape on part of them or something to try to tease apart local versus diffusible signals? Kind of relating to your first story a little bit.
No, we haven't. I did think about, if there's a way we could synthesize like a similar scar structure and like try to, put a little band-aid in there to protect. But we haven't tried that yet. But if we could maybe get an engineer to help us, create a scar or put black tape or whatever you're recommending, then I'd be open to trying that, definitely.
Yeah, I'm sure there's a way.
Everything's possible in flies, right? So.